Thank you, Nimita. Now I welcome Dr. Kalpana Devi, ma'am, Associate Professor and Head, Department of Chemistry, for the welcome address. Ma'am, please.
A very good morning and a warm welcome to all of you to the international webinar on anti-drugs for COVID-19 and dye-sensitized solar cells. With plentiful blessings of the President of our college, Dr. M.R. Chami sir, and our Madam Secretary and Director, Dr. C.A. Vasuki, we are going to organize this webinar today. At the outset, I would like to take this opportunity in welcoming the resource person, Dr. Thanigai Malai Pillayar, Independent Group Leader, Institute of Pharmacy, Tübingen Center for Academic Drug Discovery, Germany, who has readily accepted our invitation to deliver a topic on anti-drugs for COVID-19. I welcome you, sir. It is also my pleasure to welcome the other resource person of today's webinar, Dr. E. Yamuna, Assistant Professor of Chemistry, Karpagam Academy of Higher Education, Coimbatore, who has a vast experience in the field of dye-sensitized solar cells. I welcome you, madam. I am really glad to render a warm welcome to our beloved principal, sir, Dr. M. Lakshmanaswamy, sir, who is a constant source of encouragement to all of us. I welcome you, sir. I am immensely pleased to welcome the participants from various institutions to this webinar. I welcome the faculty members of our department who always render utmost cooperation and teamwork. I congratulate Dr. A. Pushpaveni for organizing this webinar. Finally, I welcome my dear students who has always a quest for learning. Once again, I welcome you all. I wish the program a grand success. Thank you for the opportunity given.
Thank you, ma'am. Now I request Dr. M. Lakshmanaswamy, sir, the Honorable Principal, Kungunadu Arts and Science College, to deliver the presidential address. Sir, please.
Thank you, Kalpana Devi, madam. Good morning.
Thank you, sir.
One and all present here. Most respected Secretary and Syndicate Member of Bharatiya University, Dr. C.A. Vasuki, madam, and resource persons for today, technical session 1 and session 2. For technical session 1, Dr. Thanigai Malai Pillayar, Independent Group Leader, Institute of Pharmacy, Pharmaceutical Medicinal Chemistry and Tübingen Center for Academic Drug Discovery, Germany. And for technical session 2, Dr. E. Yamuna, Assistant Professor, Department of Chemistry, Karpagam Academy of Higher Education, Ichanari, Coimbatore. Head of the department, Dr. K. Kalpana Devi, and Organizing Secretary, Dr. A. Pushpaveni, Dean Academics, Dr. S. N. Madan Shankar, faculty members, research scholars, PG and UG students, and other participants. On behalf of the management, welcome you all for the international webinar on anti-drug for COVID-19 and dye-sensitized solar cells. I really appreciate the chemistry department of PG, faculty members and research scholars, UG and PG students for the selection of the excellent topic. Coming to the subject, the World Health Organization called the outbreak of coronavirus infectious disease a public health emergency of international concern. According to the WHO, Center for Disease Control and Prevention (CDC) and the US Food and Drug Administration, currently there are no medicines or vaccines that have been claimed to be useful in the prevention or treatment of COVID-19. Several existing antiviral drugs previously developed or used as treatment for severe acute respiratory syndrome, that is SARS, Middle East respiratory syndrome, human immunodeficiency virus HIV, and malaria are being investigated as COVID-19 treatments, and some of them are being used in clinical trials. According to the CDC and Chinese treatment guidelines for COVID-19, chloroquine, hydroxychloroquine, lopinavir, and ritonavir, and one of the investigational agents, remdesivir, are recommended in clinically ill older patients. The use of other potential drugs reported in different studies may be considered if treatment with the first line drug is ineffective. There are currently no complete data available from large randomized clinical trials, that are RCT, to provide clinical guidance on the use, dosing, or duration to validate the effective role, safety profile, and adverse effects on all of the trial drugs for prophylaxis or treatment of COVID 19. Until now, it is still unclear which drug can successfully fight against the disease. Therefore, for the better safety of patients with COVID 19, further clinical trials and large randomized controlled studies are needed to validate the effective role safety profile, and adverse effect on all the potential drugs, such as measure requires action at the global level. Recently, novel coronavirus COVID 19 identified as one of the acute respiratory syndrome coronavirus SARS-CoV-2 and emerged as a pandemic disease in Asia and Europe countries in 2020. The World Health Organization has declared the current outbreak as a global public health crisis due to the variability in the amino acid and amino acid sequences. It does not develop suitable vaccines against the viral protein. Hence, the inhibitor to be developed against the viral protein for the coronavirus is a promising idea to develop structure-based drugs from the photochemical compounds. Here, the novel drug was identified and well studied against the viral receptors by using the molecular docking techniques. Phytocompound 6-gingerol possesses excellent drug like lines with zero violations and very good pharmacokinetic properties with the highest binding affinity ranging from 2.8764 kg per mole to 15.7591 kg per mole with various COVID-19 viral protein targets. Our, so with this few introduction, I'm also eagerly waiting for our guests from Germany and the guests from the HRI Coimbatore. Uh, Really, once again, on behalf of the management, I appreciate the chemistry department, PG and research department of chemistry for selection of the wonderful topic. I'm also eagerly waiting for the chief guest speech. With this few introduction, thank you for the opportunity given to me. I wish the program a grand success. Thank you.
Thank you, sir. I now welcome Dr. Amutavalli ma'am, Assistant Professor, for introducing the resource person of the first section, Dr. Tanigi Malai Pillayar. Ma'am.
A very good morning to one and all present here. It's my great honor and pleasure to introduce Dr. Tanigi Malai Pillayar, Independent Research Group Leader, Pharmaceutical Chemistry, Institute of Pharmacy, University of Tübingen, Germany. He completed his PhD in Synthetic Medicinal Chemistry from Chungnam National University, South Korea in 2011 and postdoc studies at Tokyo University of Pharmacy and Life Science, Tokyo, Japan, from 2011 to 13. His current research focus is on total synthesis of natural bioactive products, methodology development for synthesizing novel organic molecules with potential biological applications, drug discovery and developments. His achievements have been recognized by many different awards, including the Young Researcher Award for 2009 to 10 from Brian Korea, 2021 Scholarship South Korea. He delivered lectures in various conferences. He has guided both masters and doctoral students during their exchange program. He has published number of journal articles in well-reputed scientific journals. He also serving as a reviewer for many international recognized journals. It is so impossible to mention his every single accomplishment because they are a lot. It is such an honor for us to have you today amongst us, sir. Thank you so much for joining with us. I now welcome Dr. Tanige Malai to take over the session. Sir, please.
Yeah, good. I hope you can hear me.
Sir.
I hope you
Yes,
can hear
sir.
me, right?
Yes,
Yes,
sir.
sir.
Yes, sir.
Yes. Thank you very much. I really good morning to everyone, and thank you so much for the introduction and the invitations. I think Dr. M. Legeshman Swami, he's already has given already a general perspective of coronavirus disease. So I don't know how I'm going to differ from him, but let me see how to point out some of the things that are happening in this situation. Yeah. So can I share my slides? Because there was a problem, but let me try once again. So can you see my slides?
Yes, sir.
Yeah, again,
Yes.
the problem is I cannot move the slide, and I cannot expand the to the full version. So, dear Pushpavani, can you please present
Yeah.
for me? Yeah,
Yes, sir.
as
Yes,
we
sir. Yes,
discussed.
sir. I
Yeah,
can
thank
take.
you very much. Let me
Okay.
stop that. Yeah.
Okay. Is my PPT is visible?
Is that the full version? I mean, the full screen.
Yes, sir.
Okay, right. Okay, then I will start now. Yeah. Okay. Thank you once again for inviting me. Yeah, you all know what is going on right now. But what I'm going to present here, I'm going to give an overview about how we are facing this situation, how the past experiences helped us to fight against the coronavirus disease caused by SARS coronavirus 2. Yeah, can you go to the next slide? Right. So I'm going to generally give the brief about the coronaviruses and their genomes, how they cause disease, life cycle, which is very important, and how the mutants affect the human effect to fight against the the coronavirus disease. So a little bit about the spike protein and then variants of SARS coronavirus 2. What are the treatment options that are available for treating the coronavirus disease? Some vaccines and antibodies that we have so far. And I'm also going to give some interesting topic about the small molecules that target the very important enzymes that are involved in the virus life cycle, mainly main protease and papain-like protease. And there is another enzyme called RNA dependent and RNA polymerase, which is also important as a drug target. And in serious case, what is meant by cytokine storm, how they induce the severe patients very, very severely. And finally, this summary. Next, please. Okay, the first, I'm going to talk very general about the coronavirus 2, SARS coronavirus 2 and their family, genome, life cycle, and different mutants. Next. Okay, the coronaviruses are generally classified according to the International Committee for Taxonomy virus. So when you see that the coronavirus belongs to the family of Coronaviridae, which has two subfamilies. One is Coronavirinae and the Torovirinae. The subfamily is further classified into 4 different genus that are alpha, beta, gamma, and delta coronaviruses. And we have here alpha and coronaviruses that are really dangerous to the humans because they transmit from animal to human and from human to human. And in generally, and you can see that the all dangerous coronaviruses that are SARS coronavirus 1, MERS coronavirus, and the current SARS coronavirus 2, they are under the beta coronaviruses. In general, the alpha and beta coronaviruses infect only humans, but delta and gamma coronaviruses infect both birds and animals. Yeah, please. Next. So far, we have discovered 7 human coronaviruses that are human coronaviruses to 2029, which was discovered nearly 70 years ago. So after that, we have been discovering new viruses. The first 4 human coronaviruses are very mild that cause 1/3 of common cold in humans in the world. The other 3 coronaviruses, that are SARS coronavirus 1, which was emerged in 2002 in China as well, though no surprise. It infected already nearly 10,000 people, but it was immediately contained before spreading to many countries, though the mortality rate was nearly 10 to 15%. In 2012, in the Middle East region, there was another virus called Middle East Respiratory Syndrome, that is MERS coronavirus, was emerged in Middle East region. If you see that the mortality rate, it was really high, but it is still infecting the people, but not as like before. And now what we are facing is SARS coronavirus 2 that gets the name because of the SARS coronavirus 1, because they have very similar structure. And this is the data about 1 week ago that it was already infected more than 200 million people, and it killed about or more than 4 million people, with the mortality rate of approximately 2% according to the World Health Organization. So that the SARS coronavirus infects the upper respiratory tract, and then immediately spread to other very sensitive tissues like lung epithelial cells and heart, kidney, brain. That's why you have this very severe respiratory problem, and in a severe case that you get the organ failure, and then you get the cardiac arrest, heart attack, and so many other very dangerous symptoms that leads to the death in a severe case. That severe case means it is nothing but production of the cytokine storm. I will talk about this a little bit later in the last part of slide. So the problem is, even if you cured from the SARS coronavirus 2, or if you have very long COVID disease, you will still have symptoms after clearance of the viruses. For example, and also you will get fatigue, headache, loss at the started small, sometimes muscle weakness, and you get always low fever. So this is not that something that when you get the coronavirus and cured, that means you will still have some problems. So how could we fight against this virus? That will be discussed in next slides. Yeah, please. Next one. Yeah. So how the structures looks like? Yeah, they all RNA viruses that are including SARS coronaviruses. They are positive sense single strand RNA virus with larger size of genome, probably from 29 to 32 kilobytes. So they encode. So the viral genome encodes for the both non-structural proteins and structural proteins. Okay, and they have like ORM, that is open reading frame 1A and 1B proteins. They will be cleaved into many different parts of the small non-structural proteins. And the other part of the open reading frame polyproteins encodes for the structural proteins that are spike protein, membrane protein, envelope protein, nucleus protein. So this is the structure of the SARS coronavirus 2, and they have a very basic structural features. So these structural proteins are not only important for giving the structure of the virus, but it is also important for reducing the human system in order to facilitate the viral replication. So the structural proteins are also very important, especially the spike protein. Yeah, I will talk about the spike protein in the next slide. Next slide, please. Okay. So in order to make a therapeutic application, it is very important to understand how the virus evolves and what is the life cycle of virus in human cells. So this is very important to understand. Then only you can probably think about to stop the virus replications. When you think about the viral replication, it is very complicated. It is not very simple to understand. But how could we understand? But we can classify 4 different important steps in the viral life cycle in human cells. The first step is the entry. That means the spike protein of the virus can bind to the receptor of the human cells, and this will be cleaved. Once it is in the cells, then it will be cleaved to produce the viral mRNA. Once you have the viral mRNA, that it has to be immediately translated by ribosome to produce the viral protein. Yeah. Once you have the viral protein, they are inactive, but it has to be cleaved by different enzymes to produce the active virus particles. And this protein is cleaved by 2 important proteases that are main protease and PL, papain-like protease. So you can target these proteases to stop the viral replications as a second and third step. And there is another step that called sorry. There is another step. It is this RNA dependent and RNA polymerases, which is the kind of catalytic part which can produce the different copies of mRNA. So once you have the different protein, unstructured proteins and duplication copies of mRNA, they all assemble together and make a new virus particles. Once they mature, and then they will exit from the cells to infect the other new cells. So once it attacks the new cells, then the cells reacts, which produces cytokines, different pro-inflammatory cytokines. When it is uncontrolled, then you have the problem of immune system failure, and that leads to the very severe cases. Yeah. So in order to develop the therapeutic agents, or in order to stop this viral replications, if you target any important step of this viral replication, then it will become a very important therapeutic approach. Then you would be able to stop the viral replication in human cells. Yeah, please, next. Yeah, so I will talk about a little bit detail about how they enter, how the virus enter into the human cells. Okay, the virus has the spike protein, as you know. Yeah, it enters into the human cells. That I show here. But how it enters? Because you have the receptor that is called the angiotensin converting receptor. So it binds. The spike protein binds to the receptor. But if it go a little bit deeper, the spike protein has 2 parts. One is S1 receptor. One is S1 and another part is S2. So the S1 is used for the receptor binding. It initiates the binding. And the S2 initiates the fusion of the spike protein. You can see here, and there is a very specific important region that is called receptor binding domain. Even if you have the spike protein, that interacts with the receptor binding domain of the receptor. So that is more important. And you have several sub-receptors or sub-enzymes, something called like furin or transmembrane serine proteases, which can help the spike protein to transmit effectively, because it balancing the spike protein, and once it is binds, then it cleaves the binding as I shown here to enter into the host membrane. So these steps are very, very important. So you can immediately think about, okay, once you stop this binding, then the viral cannot enter. So that will become an important approach. So this is how the vaccine is developing. So we will talk about little in next slides about this vaccine development. Please, next slide. Okay, so that was very important. As I told in the previous slide, that you could stop the virus replication by just inhibiting the virus entry. But what is the problem? The problem is you will always have different mutations on the virus protein. If you target the spike protein, that is fine, we can stop. But the problem is you will always have the changes in virus spike protein that will many times facilitate the virus replication. For example, we have different variants, alpha, beta, gamma, and delta, and they have different properties. For example, alpha variants, they have different mutations in different region of the spike protein, which actually this mutation facilitate the more than 50 to 70% more infectious than the original virus. Beta variation, that is called beta variants, which was found in South Africa. It was about 50% more infectious when compared to the original virus. But what is the dangerous one that we are facing now is the Delta variants, which was found in India. And people and the scientists believed that the second wave is supposed to be the cause of the Delta variants, because it is not very effective. The vaccines that we are having are not very effective about the Delta variants because they have a mutation in the very important region of the spike protein, that is in the receptor binding domain. So the mutation is actually facilitate the binding of the spike protein to the receptor. So it even facilitate the binding of the spike protein, which enhance the spreading of the virus. And also there is a mutation in the very important region, that's the furin cleavage site. The furin is the enzyme which is important to cleave the spike protein to enter into the cells. So they have very critical mutations in the very important regions, which actually facilitate the virus spreading. That is the reason why that the world had a second wave, which was very severe and with a lot of deaths. So these are the problems with variants that you are facing now. Yes, please, next slide. So I talked a little bit about the virus, what is virus and their family and genomes, and how could we target and the replication. The next topic is we understand something about the viruses and how they behave. But the next, how could we treat the virus infection, which is caused by the SARS coronavirus 2 and that causes COVID-19? There are two options. One is preventive. We can just prevent before the virus attacks us. The next one, the treatment option is after the infections. Okay, once you get the infections, then how could you treat? So we have two treatment options. One is the preventive, and another one is treatment options. The preventive is mainly targeting the spike protein. As I said, if you target the spike protein, you can stop spreading the virus. So how could we target? They are mostly biologics, that is vaccines. We have so many different vaccines now, like that are based on mRNA, vector-based, even protein vaccines, and the inactivated viruses. This is from China, and the BioNTech and Moderna from US, Pfizer. And the treatment options that you can treat you can target the specific enzyme which are involved in the virus replication. So you can actually target the virus, but here the virus after the infections. Yeah, and we have like antibodies, small molecules approaches, and also targeting the immune system. And there is a treatment option for the long COVID-19. So I'm going to talk a little bit, or maybe the overview about these topics. Yeah, next slide. So the first topic is SARS coronavirus 2, and how it could be targeted by vaccine, antibodies, and small molecules. Please, next slide. Yeah, this is something kind of a general overview about the vaccines. That what are the vaccines we are having? We are having so many different kind of vaccines. Some vaccines can be used by some vaccines contain inactivated virus that can be injected into the human cells, and the virus can produce a spike protein, and the spike protein, once you have the spike protein, then the human immune system will become activated, and it will start fighting to the against the spike protein. So this is how it gets the practice. This is how it gets the practice, but when you have the real COVID virus, then it can kill the virus. So this is the basic concept. But different companies uses different techniques. Yeah, some companies, for example, the company like Sinovac from China, they use adenoviruses, inactivated adenoviruses. But BioNTech and the Moderna virus, they use mRNA based, which is very modern and which is very new, and which is the most powerful that have been claimed, which has been claimed to be that it shows more than 98% effective against the SARS coronaviruses. But probably against the Delta variants, it is not even less than 80%. So this is the most effective vaccine that we have so far. And there is another virus from Covishield from AstraZeneca and Johnson & Johnson's Sputnik virus. They use the vector virus to transmit the virus into the cells, and then that can be produced. Then the virus can produce the spike protein, and the spike protein, and the immune system will fight against the inactivated spike protein to get the practice and to become ready when the new virus and the real virus comes into the cell, so that it can destroy the coronaviruses. And some companies, they use viral protein directly by injecting inactivated viral protein, spike protein. And some companies, they use just virus-like particles. So there are different techniques have been used for the vaccine preparations, and each one has a different effective and different techniques. But BioNTech is the best so far that we have, which is very new and a modern technique based on the mRNA. Please, next. Yes. So in the previous slides, I talked about the vaccines, but we have also now options that can be used, that are monoclonal antibodies against the SARS coronaviruses. So during this pandemic, there are a lot of companies that have developed a series of monoclonal antibodies, which is mainly for the patients with the mild to moderate disease, or even people at the high risk of severe progressions. So several different monoclonal antibodies have been authorized in an emergency based, because we have no real treatment option. So some of they have been approved by FDA, by the United States of America. And the main target of this monoclonal antibodies is, of course, is spike protein, and mainly they target this receptor binding domains. Yeah. So once you block the receptor binding domain by using the monoclonal antibody, then you will stop the entering of virus into the human cells. So that is the basic concept. And we have some monoclonal antibodies that are already approved by FDA. The one with casivimab, the map indicates the monoclonal antibody. So you get always the name with MAB, that is mean monoclonal antibody. And there is another antibodies which is prepared by the Eli Lilly company, which was approved in February 2021. And another one is sotrovimab, yeah, which was also approved for treating the coronavirus infections, mainly for the people with moderate disease. Yeah. The next one. Next slide, please. Yes, the next target would be the treatment options, not the preventive based, but the treatment options. Okay, but the problem with vaccine and the monoclonal antibodies are, you will get, with the vaccine, you will always get some resistance, because of the mutation of the spike protein. So it is not always effective against the virus. That means you have to look for another option to treat the virus. Okay. What do you think about the monoclonal antibodies? The problem is they have very severe side effects by using the monoclonal antibodies. That is one point. Another one is the production cost. Another one is the storage problem. So you have it is not easy to store, because you really need very low temperatures, like -80, -80, -70. So it is also for the vaccines, some vaccines. Yeah. So it is not easy to transfer from one place to another place. So it is really very cost effective. And the problem is, of course, you have a very narrow therapeutic index. and a severe side effect. So ultimately, that means you need to look for something else which is more important, which is more effective. So okay, that is the treatment options. Okay, once you get the treatment, so how you could get the how you could cure the virus infections. So we have two options here that we can target using the small molecules against the viral proteins, against the viral proteins and against the host human, host proteins that has been used by the virus for the virus applications. And in a severe case, you have the different options that how to overcome the immune system. You have the anti-immune drugs that can be used by small molecules and even antibodies. And I will talk about treatment of long COVID-19 in the last part. Please, next slide. Okay. Okay, so okay, you want to target the viral proteases, the viral replications. But the problem is, when you have the pandemic situations like this, you cannot really develop the drugs very immediately because the drug development process takes nearly 5 to 10 years. But then how could you develop a new drug? Or how could you develop a therapeutic drug against such a pandemic disease? One very, very simple, I cannot say simple, but one brilliant approach is the drug repurposing. What is meant by drug repurposing? You can repurpose the drug that has been developed for another diseases with similar therapeutic, with a similar disease manner. For example, we have now SARS coronavirus 2, but we have been repurposing the drugs that have been developed for the virus, for example, HIV and hepatitis C virus. So we have been testing the compound called lopinavir and ritonavir that were approved for treating these human coronavirus deficiency viruses, HIV. And it was effective. It was effective in vitro studies, but the further development was not continued. So we can utilize the drugs that have been developed for the other purposes. And the other option is we can also use drugs that are in the clinical development for other disease. And we can take the drugs that are stopped for safety reason, not for some other reason. For example, efficacy and economic consideration. Once it is very expensive to make drug, they might stop. But in a situation like this pandemic, probably you could try those drugs and test if they are effective. If they are effective, it is really useful treating the viral infectious disease. And there is not the option that the drugs developed for clinical development, but not started for different reasons. Obviously not because of safety, because safety is more important, but it might have stopped for some other reason, like I said, in the economic consideration or some other purpose. What is the advantage of using the repurposing? The advantage is you can develop the drug very fast, and you can reduce the risk of failure due to the safety. Because if you want to initiate a drug discovery program, only 10 or even 5% out of 100% could succeed and could reach to the market. That means the 90% or 95% are failure. But if you have already the drugs which are developed for other reason, so you don't have a reason to failure those drugs because you know everything, the safety property and pharmacokinetic properties. The other problem is the reduced cost, because you already made it and you know everything about this drug, so you don't have to invest billions of dollars, and which is also not useful in the pandemic situation. Okay, that is the drug repurposing approaches. That is very good that we have been practicing with many different drugs, and some are already in a good position, and some are already in clinical trials because they show very good results. What is the other option for the long-term goal? The long-term goal is to develop a new drug. That means you have to start the drug discovery from the scratch. You can target the viral, preferentially enzymatic, which are critical for the viral replications. In this case, this is the proteases, polymerases. We will talk about this. So the advantage is you can specifically target the enzyme. You are not simply injecting the drugs in the cells, which can kill anything. But here, you know the target, what is target, and you can specifically target those enzymes or receptors because you know that. And so the treatment will be very specific. Once you have this very specific target, then you will have the side effect, less side effects. But the problem is the problem is you can develop the resistance. Yeah, once you target this one, then the enzyme will start developing the mutation. Then probably your drug will not be effective. So you will get the narrow therapeutic index. So this is the disadvantage when you target the viral target. We have another option that you can target the host, that is the human, because the virus infects the humans. So we can target the human cells. In this case, you would not develop the resistant development. But the other problem is the side effect, because when you target the host proteins, the problem is if the normal process, you will have the interference with the normal process, because if our ribosomes produce the protein, if the virus hijacks our ribosomes, then it can produce its viral protein, not the human viral, not the human protein, which is important for the normal process. So this is also the disadvantage with the by targeting the host target. Yeah. Next one, please. Yes, here I was talking about the vaccine, which that targets the spike protein or the angiotensin converting enzyme receptor. The vaccines mainly target the spike protein, but here I'm talking about the angiotensin converting enzyme, which is the receptor from the human. Yeah. So during the initial of the pandemic, there have been so many reports and very exciting data about some small molecules, especially the anti-malarial drugs like chloroquines and hydroxychloroquine. It was a kind of super news every day that it cures they cure the SARS coronavirus infections. But the basic concept was that the chloroquine drugs, I mean anti-malarial drugs, they are basic. For the viral entry needs the acidic pH value in the endosomal region. But the chloroquine is a basic, so it increases the pH value of the endosomal pH, which can ultimately stop the virus to release its viral RNA. So this is the basic mechanism that how chloroquine works. But there are also some other mechanisms that in different parts of the viral replications. So based on this data, they have started more than 100 clinical trials around the world, but unfortunately none of them were confirming that they are really effective. And we had a very controversial clinical research. So ultimately the World Health Organization did not recommend to take the chloroquine, because some people, they were already started taking the chloroquine analog, which has been used for the fish tank. I mean, people already started taking those kind of drugs, which makes more severe than the normal coronavirus infection. So there was a lot of controversial and different problems with anti-malarial drugs, but it is ultimately it's not the solution to treat the coronavirus. And there is not a small molecule, that is umifenavir, which has been already approved for the influenza virus, I guess, and that also showed some moderate activity, and then some clinical trials were already started, but there was no good clinical results, and scientists suggest that it might not improve the prognosis or accelerate the SARS coronavirus clearance, even if you take this. Another drug is Comestat, which is a serine protease inhibitor to block the viral entry and reduce the infections. And antiviral activity of this compound was already proved in Kalu cells, Kalu lung cells, which is one of the cells that is very sensitive to the SARS coronavirus infections. So the Comestat is already approved in Japan for the treatment of chronic pancreatic associated pain. And there have been many clinical trials, and recently the phase 2 and phase 3 clinical trials are undergoing in USA and Japan, but we don't have any update about the outcome of the research. Probably we will see something about this track in the future, near future. Yeah, next slides, please. Okay, so this in the previous slides, I was targeting the angiotensin converting enzyme for the virus entries purpose. So okay, once you have the virus inside the cells, but still you can cure the disease by targeting two important proteases. That is main protease. We shortly call it as MPro, and papain-like protease. We call it as PLPro. So we're going to talk what is the role of these enzymes, and how we could target these enzymes. Please, next slide. Yeah, if you see this the replication cycle, you see that the virus enters and then leaves to produce the viral mRNA. Once you have the viral mRNA, it can be immediately translated into protein, viral protein. Okay, so once you have the viral protein, they are not functionally important, so it has to be cleaved or processed into different parts. And this protein can be cleaved by this protein can be cleaved by two proteins. One is CL-Pro, that is otherwise known as MPro, as I said before, and another one is papain-like protease. And if you just look at the right side, once you have the protein here, and the first three part of the polyprotein can be cleaved by the papain-like protease. But if the other enzyme, that is main protease, that cleaves the polyproteins at not less than the 11 conserved sites. So the major portion of the proteins can be cleaved by the main protease. Even those two proteases are very important, but I would say main protease is even more important because it takes a larger portion of the viral protease, and not only because of that, the protein cleavage by main protease it also gives the another important enzyme that is called RNA dependent RNA polymerase. That is very important for producing the viral mRNA copies. So in both sense, this mRNA, the main protease is very important even than the papain-like proteases. But of course, the both proteases are playing very important role in cleaving the viral proteins that are important for making the viral replication complex, and that makes the new virus particle to produce the new virus, and then affects new cells. So in that sense, these both proteases are very important for the viral replication. So we will talk about how are we going to target this one. Yeah, next one, please. Okay, when you just go into the molecular level, yeah, when you just go to the molecular level, then the main protease that looks like this one, because there have been we have a lot of X-ray crystal structure of this main protease. Just when you just look at the left side, which is very complicated, but you can just have a look at the right side of the picture, because this main protease is a dimer, blue and red color. This is a dimeric form, and the dimeric form is very important for the activity. And you have the catalytic diode, which digests the peptide substrate. So that is very important. And I also highlighted here in the red color in the left side of the part and indicated as the catalytic diode. So this is the catalytic point, which is very important. And this protein is highly conserved among all coronaviruses, and it shares more than 96% similarity to the other SARS coronavirus proteases, which was emerged in 2002. So And as I said in the previous slide, it is also important to produce the function of polymerases, and the helicase. They are also important in the replication cycles. So in sense, it is really important, and also it's important to know how it functions. So we have published a lot of papers regarding review and research article about this protease and its inhibitors. Probably you could have a look at some of these references. And mostly when they target this protease, they inspired basically from the natural substrate, and just by changing the natural substrate, they designed the new inhibitors. Mainly, the main protease as the very sensitive, and it cleaves the amino acids, that is glutamine, as I shown here. So this glutamine can be converted into different moiety, and this is one, that is lactam, which is an isomer of isoster of this glutamine. But so by using this one, probably you could stop cleaving the main protease. So by that way, you can stop the peptide substrate cleavage, and you could stop the production of small non-structural proteins, which are important for the viral replication complex. So this is the basic mechanism, how the main protease functions, and what is the role of the main protease. And if you just look into the deeper, then you could already see the 3D blueprint of the main protease. So by having this information, so we could develop new inhibitors. Yes, please, next slide. Yeah, this is very general information about the assay, that because they have general information about the assay, that how they determine the IC50 values of the inhibitors. This is the basic concept, that they use kind of fluorescent substrate which mimics the natural peptide substrate, and they have both sides the fluorescence. Once in the normal process, when the main protease is cleaves here, and you have two fluorescence, and then it becomes, and then and these activations can be quenched by using the other fluorescence. But when you have the inhibitor, this fluorescence just goes part away, and once you have the fluorescence, and then you get the light, the fluorescence excitation energy by using the 490 nanometer, and this fluorescence energy can be directionally proportional to the inhibition of the protease. So this is the basic concept, that how they determine the viral the inhibitory activity of proteases. This is very basic. I just wanted to show some. Yeah, please, next. Yeah, in the previous slides, that was the in vitro enzymatic assay. That was not the problem, but every lab can do. But in order to see whether your inhibitor shows an antiviral activity, that means you have to use the cells, and this, that means you have to use the viral infected cells. Yeah, and you need a special protected laboratory, which means that you have to get this special permission from the government. So it is not to just check the antiviral activity of the virus like SARS coronavirus 2. But we have some requirements what the cells need to have. That means the cells has to express the angiotensin converting enzyme for the entry, which is important because the ACE2 is the receptor for the SARS coronavirus 2. And also you need the protease to cleave the spike protein, that are TMRSS2 and furin. And the cells that you are choosing, that should reflect actual infection of your cells. Yeah, these are the basic three requirements. So what are the cells have been used actually? Means the Vero cells, cell line expressing the AC2. So we are people are using the Vero cells that has been got from the African green monkey kidney cells, which is expressing highly the AC2 receptor, and also the lung cells, and which also express the AC2 and the other two sub proteins like transmembrane serine proteases and the furin. And scientists have also been using the genetically modified cells, like from the epithelial cells and also from the on the viro cells, they have some gene techniques, but the thing is they have to express the receptor and the enzymes that have been used for cleaving these spike proteins. So these are the basic things when you're choosing the cells for testing the antiviral activity of your compound. So these are two basic things in the discovery of your anti-SARS coronavirus antiviral drugs for the SARS coronavirus 2 or any coronavirus that we need to know about the things. Next, yeah. So once I tested the compound, that then we have so many different inhibitors. As I said in the beginning, the primary inhibitors are mainly based on the peptide-derived inhibitors that are inspired from the natural peptide substrate. So they take the natural peptides and change something, and that will become drugs. I mean the inhibitors. Yeah, I will show you how we have a lot of inhibitors. The next slides. Yeah. Okay. So when you consider about the peptide structures, there was a drug called upentavir, which has been used for the rhinovirus infections. Yeah, rhinovirus also is a virus which has the very similarity to the SARS coronavirus 2 and other coronaviruses as well. So other coronaviruses as well. So they took this structure, which was actually inactive for the SARS coronavirus 1. sorry. Also for the SARS coronavirus 2. So what they did, they changed this position. If you see here, then I indicated like they changed, I indicated with red color. They keep everything same with constant, but they change at this position. We call it this one as a warhead group, because we always attach very electrophilic reactive group which can be reacted with the active site of the main protease. Yeah, in the active sites we have, if you see here in the left corner downside, then I showed here that the actual packet of the main protease, where you have the S1 prime, that is S means a sub packet. Yeah, so the peptide binds in different pockets of the protease, and they have the preference. And in the catalytic site, you have the cysteine 145, which is the nucleophile. So once you have, that means if you have the nucleophile, then ultimately you have to design something with electrophile. Yeah, then only it can be like can be reactive. So we have very different approaches here. That means that this warhead group can be designed by using different reactive groups that are Michael acceptor. So that is very important, very sensitive to the nucleophile. Michael acceptor, of course, aldehyde, some active reactive ketone, and the ketomine. So we scientists have utilized different reactive groups by targeting this cysteine nucleophile. So here, so as a first example, the scientists have recently designed something like this peptide-derived structure, where you have the aldehyde. This is the aldehyde, which is very reactive. So what happens, this cysteine molecule reacts with aldehyde to make the like a ketone structure, which is very stable. And then the protease cannot proceed further because it cannot kill you, and ultimately the process of protease will be stopped. So this is how the people develop the drugs, and this is the example of aldehydes. And two examples, they are really effective, very effective against the SARS coronavirus 2, and also showed antiviral activity in cells without significant cytotoxicity to the cell. So because it's very important also to consider when you're testing your compound for the antiviral activity, if the compound kills your cells, it doesn't mean that it shows antiviral activity. It could be because of the toxicity, your cells will be killed. So you have to be really careful when you're testing the antiviral activity of your compound, whether it kills your cells or whether it kills the virus. So this is more important. Yeah, please, next slide. Yeah, this is another class of drug, that is alpha ketamide. that is alpha ketamide. This is the simple peptidic-derived structure. I also indicated here P1, P2, P3, and P1 prime. P1 prime is always is a warhead group that is the reactive group that I told, which can be or which should be very reactive, and you have the different residues for the different pockets. And what happened during the pandemic of SARS coronavirus 2, Shang et al. They took an old drug which has been developed for SARS coronavirus 1, and they tested against the SARS coronavirus 2 main protease because they have a very similar structure, and it was really active. And then they modified systematically to produce some very effective inhibitors, and you could see that it is already in a good range. And then they further modified to get a very potent compound, and this inhibitors is like a broad spectral antiviral compound. That means it shows activity against different coronaviruses. Against SARS coronavirus 2 main protease, SARS coronavirus 1 main protease, and also for the main coronavirus MES coronaviruses, because they are all the coronaviruses, and the protease structure is almost similar to each other. So it is not surprised that it shows the multi spectral activity. And I think this compound is already patented, and also it's going for the clinical trials for testing. But we do not know anything about this drug, any update so far after the first report in the Science paper. And this is kind of the pioneer research paper which was published in the early of the SARS coronavirus pandemic. So that means it's very important, and many researchers have been inspired to design some different inhibitors. Yes, next one. Well, yeah, this is most interesting for us because this work is done by us, which was mainly in Japan. And this work is actually started even before me, in the range of 2005 or 2006, something like this. But what they did, this is the natural peptide substrate which can be digested. Yeah, the arrow mark indicates the cleavage site where you have the glutamine here, and the main protease cleaves. So what we did, we just took this part from the left part from P1 to P5. We designed and we just attached lactam instead of the typical glutamine. Yeah, and then we attach some different ketone. This is the example of keto derivative, and we attach the thiazole. And we tested this compound which was which was in the moderate range. It was like the binding affinity of it was like 2.2 micromolar, which was not very strong. What we did, we docked this compound into this protease structure, and we just scanned the binding pose of the inhibitor. But we found that in this warhead position, that is P1 prime position, we saw that there is some space where you can introduce some bulky group and see whether it increases the inhibitory activity or not. And also we found that at the P4 region, the phenyl group is like looking outside of the packet. So we thought that maybe we can just rigidify this fold and see the activity of this compound. So this was a basic prediction to design the new inhibitors, and then we studied the structure-active relationship of the lead compound. Next slide. Yes. So we and if you see more closely, this is the active structure, how they look like, and you have the cysteine nucleophile. So it is very clear that you have to target the cysteine nucleophile. What we did, we also introduced a trifluoromethyl ketone by using the different, the same residues which are same as in the substrate. Yeah, and this is the binding packet. After having very serious or many efforts, that we synthesize this compound with the benzothiazole, here, and to our surprise, the active was really improved a lot from I mean, 135,000 nanomolar to just 4.1 nanomolar. It was so effective. It was so effective about the SARS coronavirus too. And and this is the kind of the peptide, tripeptide. I actually, when I was in Tokyo, my topic was to reduce the peptide bond, to reduce the peptide to a small molecule, like, I was there only for 2 years, and my topic was I want to make from peptide to small molecules, because the peptides have always problem that it can be easily cleaved or hydrolyzed when you in the cells, in the physiological concentration. So my topic was actually to reduce the peptidic bond. So what I did from tripeptide to dipeptidic nature. So we were the one to introduce the indole moiety, 4-methoxy-indole moiety, which was found to be one of the best moiety at this position. And we could even maintain the activity by using the dipeptide. The dipeptide was about 4 nanomolar, and the dipeptide was 6 nanomolar, which was almost same. So what we did during this, when this pandemic happened, we resynthesized those compound 1, 2, 3. We resynthesized those compounds that have been already reported for the SARS coronavirus 1. We reported this work in 2013. We resynthesized those compounds, and we also tested now at SARS coronavirus main protease 2, and we also found that they have very similar activity about the SARS coronavirus 2, and this is the typical binding pose, how they inhibit. You see that this is the active keto group here, and the nucleophile attacks the carbonyl group to make the keto structure, which is stable. So that means the cleavage of the protease is stopped. So this is the concept. And this work, and we also studied further things like pharmacodynamics and oral bioavailability, but we took this compound which has the indole moiety here, YH53. We studied the oral availability and also the preclinical studies of this compound. But although the results were not that great, but it is very important that it could serve as a lead compound which can be used for developing the real effective drugs. So we recently published this work in the Journal of Medicinal Chemistry. Probably you could see, and or if you're interested, I can send you the paper if you want. But they are very we have a very inspiring story about developing this compound. The next one. Next slide, please. Okay, there is another peptide drugs. Can you see? As you see here, they have 4-methoxy-indole as well. But the problem is, it was developed by the company Pfizer, and they reported only in the patents. What is meant by patent? Probably you could not see the full structure of the compound sometimes. Okay, so me as an academic researcher, I developed a drug with 4-methoxy-indole independently, and the company, as always, they do in a secret way. They develop compounds with 4-methoxy-indole, and what is the difference we have between me and Pfizer? The only difference is here at this warhead position. They have the alcohol, methanol. I have the benzoyl. So that was the only difference. We didn't know that they have the compound like this, but once we have the SARS coronavirus 2 infection, then the company started reporting the compounds like this. This is one example which showed very good activity against the SARS coronavirus main protease. As you see that they are in the low nanomolar activity, and they also showed broad spectral activity against the different SARS coronaviruses. And very importantly, they are very selective against versus SARS coronavirus 2. That is also more important thing. And they recently published the work, the clinical trial of this compound. Can you just go next slide? Can you go to the next slide? Okay. But the problem with these inhibitors, they are not very much oral available of this compound. They are not very much oral available of this compound. And another problem is they have a problem in reaching the targets. So what they did, they synthesized prodrug, the phosphate prodrug, and then they introduced by intravenously to target the main protease. The basic concept is this: the phosphate can be cleaved in the physiological concentration of the cells, and then it can become an active. And this is an active a molecule. But the problem is it has the very low oral bioavailability, and the other problem is you have to infusion. I mean, you have to infuse this drug about 500 milligram, which is very high level, for 24 hours. So that means when you have the patients in a serious condition at the hospital, probably you cannot force them to inject the drugs 500 milligram, which is really high, and for 24 hours, so practically, which is not possible. And recently there were a kind of article. They asked me about the opinion of this compound. I was severely criticizing about this drug, that it is not possible to have 500 milligram. And it is not also orally available. You have to inject by intravenously, and it is very painful, and it is not practically possible. And even what they did, the company as well. The next one, please. Next slide. Yeah. So that was the problem of those drugs, that they are not orally available. But we have also some examples that are orally available. The one is the boceprevir, which is actually targeting the serine protease, but SARS coronavirus main protease is a cysteine protease, but it was still active. And this drug was already approved for treating this hepatitis C virus, which is and one more good thing is it's orally available. Though the enzyme inhibitory activities is only moderate, and also the antiviral activity is moderate. But one good thing is that it is not cytotoxic, and it is already approved for the hepatitis C virus, and which is and orally bioavailable. I think they are doing some preclinical trials with this compound. The other promising candidate is the PF derivative, which is also developed by Pfizer. Okay, as I showed in the previous one, there was the previous slide, the compound that had problem of oral availability. Then what the Pfizer company did, they stopped the process, and then they developed a new drug from scratch, and very fast, that they could reach phase 1B level for treating the COVID diseases. And the most exciting thing about this compound is it's orally bioavailable. And this compound was recently revealed in the ACS meeting, and they promised probably they could bring it to the market if everything goes well. But we do not have any information about this drug. But this is one of the most promising candidates, which can expect in the end of the year, probably in the market, then if everything goes well. So these are the most promising results about the oral available SARS coronavirus 2 main protease inhibitors that shows also the very good antiviral activity. The next one, yeah. Next slide. Yeah, so far I was discussing about the peptidic drugs. Even though they are very, very, very effective and very active, the problem with peptides, that you have always problem of side effects, and because it can be cleaved into different fragments. Basically, you have the peptide bonds, so you have a lot of enzymes that can cleave the peptide bond. So that means you will have a lot of side effects, and probably you will have very low oral bioavailability. So what is the ultimate option is this small molecule. They are in a molecular size, they are very small, less than 500 gram per mole, and they have very little hydrogen bond acceptor and donor, and they must be very stable. They recently they discovered compound 223R from the lead compound ML188, yeah, which was actually reported for SARS coronavirus 1 main protease, but they were not it was not active in cell assay. But they synthesized a series of derivatives by using the UGI library, UGI reactions. That is basically you can combine different partners of the reactions and generate library of compounds, and you can immediately test those there, and you can select if something is active, then you can select only those and then and characterize to see the structure of the compound. So this is the basic concept against the UV library inspired drug discovery program. So this is also selective as other cathepsin in cysteine proteases, which is also very good and shows a good antiviral activity. This is one good example of the small molecule. Yeah, next one. Yeah, the other one is of course from our work. So we designed the compound. By using the pyridyl ester, 3-chloropyridyl ester. So we thought that it should be very sensitive to the enzyme. The basic concept is you have the catalytic site, histidine and cysteine. Histidine will behave as a base. Yeah, once you have the base, then the base can take the hydrogen from the cysteine, and then the cysteine will become a nucleophilic, and the nucleophile will attack at the carbonyl position of the ester. So this is the concept that we decide. Then you have the enzyme, acyl enzyme complex in the system. So this is the concept that we designed, and we use the 3-chloropyridyl as an active part of the molecule, and we synthesize series of compounds with the indole, benzofuran, and the benzothiazole. Yeah, we thought that the indole is very important for the activity, and we synthesize the compound with the second 2 position of the indole. Once we have the 2 position, we started investigating the position of the attachment of the 3-chloropyridinol in different position of the indole, for example 3, 4, 5, 6, 7, to see what is more important for the activity. So when you think about the structure active relationship, we found that the second position that I showed here, which was more potent than other, and this is the order, like the second after comes 7, 4, and 6, and then the 3 position was really worse, which was almost inactive, with the IC50 value of 21 micromolar. And another important interesting thing, that this NH of the indole can also be replaced by oxygen and sulfur, and that was even very active when compared to the indole. For example, the indole showed like 24 nanomolar, but the benzothiophene showed 26, and even with the benzothiazole, we reached up to 6 nanomolar, which was equal to the peptide that we developed. But the advantage is a small molecule, and we published this work very recently in the Angiovendochemie. I probably some of you know about this the journal, which has the more than nearly 15 impact factor. So which was really very good work, and we are still continually working on kind of same series. But the problem is it is not published, so it's not possible to show the structure. Yeah, can you see the go to the next slide, please? Okay, so far I was talking about the main protease inhibitors that are peptide based and the small molecule based. And as I mentioned in the previous very early, that we have 2 proteases. One is main proteases that we finished now, and we have another protease that is called the papain-like protease, which actually cleaves the viral polyproteins, only the first 3 position of the polyproteins. Okay, it has a small portion of the polyproteins, but which is also important for formation of the replication complex, and it has also the direct link with the immune system. So in that sense, it is also important for the activity. Are you trying to make a full screen?
Yes, sir.
Yes. I'll wait. Please give enable editing, yeah? Yeah, thank you. Yeah, let me continue, yes. So, this is so the papain-like protease also is very important for the viral replications. But when you compare it with main protease is even more important, and the extensive research is already published. But here, when considering the SARS coronavirus 2, there are only few reports about targeting the papain-like proteases. And these are some examples, which is actually also based on the SARS coronavirus 1 PL-Pro inhibitor. This is the compound which is in the left side, GRL 0617. So they took this compound, which was already published for SARS coronavirus 1 PL-Pro, and they also tested for the SARS coronavirus 2. And they developed some series of compounds, and some of them showed a moderate even as good antiviral activity. And they recently established this X-ray structure of the PL-Pro. But still many are undergoing, but we don't have many examples of papain-like protease inhibitors when compared to main protease inhibitors. Yeah, next one, please. Okay, so this is the third topic, that is RNA dependent and RNA polymerase inhibitors. Yeah, as I said in the beginning, they are this polymerase is important to produce the new copies of. Here, once you have the viral protein, then it has to produce the viral mRNA copies for the next round. So in that sense, this RNA dependent and RNA polymerases produce the new RNA copies from the available template of RNA copies. Because this enzyme is really important in all RNA viruses, because it has to produce the RNA. And also the very important thing is that it has the very high conservations among all different RNA viruses, coronaviruses. So that means if you target one virus, probably it can also be effective on other viruses. So it would be a good approach. It would be a good approach to target the RNA dependent and RNA polymerases, and it shares like more than 95% sequence identity to the SARS coronaviruses. So one can easily target this virus to stop the viral replication. The next one. Next slide. So in order to target these RNA polymerases, there have been huge effort have been taken. And then the drugs that have been developed for different purposes have been utilized to target this RNA dependent and polymerases. These are some examples, like the favipiravir, which is already approved in Japan for the treatment of influenza, and which is also approved for treating the COVID in India and countries like Russia. And we have some other examples like molnupiravir, which was actually developed by Emory University from the academic scientists, and which is actually developed for treating the influenza, but the target is RNA dependent and polymerases. But it is in the clinical trial of phase 3 for COVID-19. And as you know, that there's a remdesivir, I think everyone should know this drug, which has been approved in an emergency based by FDA for treating the coronavirus infection. And there are some other derivatives which are basically nucleoside analogs and uridine, and they were also utilized, have been tested. They have also been tested for treating the SARS coronavirus disease, and and many are in the phase clinical trials. So hopefully we could see some results in the future. Next one. Okay, so this is something about the how the drugs work. Yeah. So I would like to give one example, that is remdesivir. Actually, remdesivir is a prodrug. It's a phosphate prodrug. Once you inject this one into the cells, and these prodrugs can be cleaved into monophosphate. So this is the concept. Once you have the monophosphate, in the presence of kinase, we have several kinases, and this catalyzes this monophosphate to produce a triphosphate. So once you have the triphosphate, this drug mimics like a substrate of polymerase. Yeah. So it mimics like a substrate as a polymerase. So what it does do, it just go and bind to the primer strand of RNA polymerase and just stick on the polymerase and stop the polymerase's activity. So this is how the remdesivir works in stopping the viral replication. So the polymerase cannot produce the duplication of RNA copies anymore, because it just stuck. The drug just struck into the primer strand of the polymerase. So this is the simple basic concept that how the remdesivir works mechanistically to stop the viral replication. Good. Next slide. Yeah, this is the last part of the presentation. So what we were talking about, they were mainly focusing on the human virus-based or human-based targeting. That means you're targeting something which is specific. Yeah. So once you have the virus, it takes some days to become very severe. Okay. To become very severe, that leads to the death. So if you are able to stop the virus in the early stages, probably you can be cured. But once you have the severe disease, then it is very difficult to treat the virus. So what is meant by very severe? Then I will show it in the next slide. Yeah, the severe means once you have the virus, once the virus infect the cells or approach the cell, the cells start fighting against the virus. That is the immune system, right? Then the cells produce a lot of cytokines, which is dangerous for the virus, which can kill. These are some pro-inflammatory cytokines. But what would happen? It can produce a cytokines uncontrolled manner. That means it will not only kill your virus, but it can also damage your normal organs, because the production of cytokine storm is uncontrolled. So this will ultimately affect your normal affect the organs like kidney, brain, heart, and the lungs. So what will happen? Ultimately your organ gets damaged, and then it will lose the functioning, the normal process, and that is the reason why you get sudden cardiac arrest and stroke and so many very severe problems, and there will be no option to cure in that case. But we have also different approaches, to treat how the cytokines can be produced, because we have several receptors or kinases that produces the cytokine. For example, we have IL-6. IL-6 is one of the key player to produce the cytokine. The IL-6 signal is very dangerous, which activate your immune system in uncontrolled manner. Then that makes a very critical problem. So, but we have already something like antibodies, like IL receptor monoclonal antibody, and we have these are like tocilizumab, sarilizumab, and cetuximab. There are different antibodies that are targeting IL-6, which is one of the dangerous signal for inducing the cytokine storm. So you can target the IL-6 to stop the cytokine storm in an appropriate manner. And these are the biological drugs, but we have also some small molecule drugs like dexamethasone, which is a corticosteroid, which is very cheaper, which could reduce 20 to 30% reductions in death of patients when they are in the ventilations and when they require oxygen. So, which is the cheapest way to treat the cytokine storm. And then there is also the kinases. Kinases are a very important player in producing the cytokine storm. So once you have the cytokine kinase, stop the kinase signaling, then probably you would be able to stop your SARS coronavirus disease. There have been a lot of kinases inhibitor, for example, ruxolitinib and sphingosine kinase 2 opaganib. They have been already tested very extensively, and some kinases inhibitors are already in the clinical trial, and they show also very effective. And probably in the future we will see the update about the status of the kinase inhibitors. But they are very important molecules. And we recently published the review article in Journal of Medicinal Chemistry that how the kinases works in stopping the viral replication, or in producing in enhancing the viral replication. The kinases are very important target, not only for the virus, but also other inflammatory related disorders and the cancers. Yeah, the next one. So this is the end of my topic. So I just like to summarize my topic or my talk, that we have discussed so many different ways how to treat the SARS coronaviruses. So we have discussed about the antiviral small molecules inhibitors to fight against the pandemic of SARS coronavirus 2. So no doubt that the vaccines have huge impact by protecting against SARS coronavirus 2, mainly mRNA and vector based. And we have also developed monoclonal antibodies, spike protein for the infected people. And we also kind of IL-6 receptor monoclonal antibodies to reduce the severity of the coronavirus disease. We also discussed the small molecule inhibitors that target the viral proteases like cysteine main proteases and PL proteases. And of course, we have a lot of examples of targeting main protease, and some drugs are already in the very advanced level. For example, the orally available compound from the Pfizer with a nitrile warhead group, which is already in a very advanced level. Probably we could hear some news about the development of the drug. But if everything goes, that will be in the market, and of course, then that will be like a treasure for us for treating the coronavirus disease. And we extensively utilized drug repurposing approaches, and we had a lot of results in treating the SARS coronavirus disease. One example is remdesivir, which was actually developed for Ebola virus, but has been utilized and tested for the SARS coronavirus 2. And of course, it is the only drug that has been approved for treating the SARS coronavirus 2. But the problem is, it is not kind of a magical game changer as we expected. It shows very minor effect and even no effect. And ultimately, the World Health Organization did not recommend, though it is approved by the FDA. So this is the controversial thing about these drugs, but it has been approved. But the report says it works for someone, and it is not working for many. So this is something that we should think about. But ultimate aim is to develop something, the globally acknowledged drugs in the future. So next slide. So thank you very much for listening my topic. I hope you could get some information, and if you have any questions, you can ask me, or you can send me by email or anything. So I'm available. So as usual, so please keep washing your hand and stay safe and keep distance. And thank you so much, and also for inviting me. And I'm very glad for this. Thanks so much. Yes, I'm done.
Thank you, sir. That was a very informative session for us in this current scenario. Now, I welcome Dr. V. Jamia ma'am, Assistant Professor, to introduce the resource person of the second session, Dr. Yamuna ma'am.
Wait a minute. Before that, audience, please ask any questions. Participants, if you have any queries.
Ma'am, I have one question, ma'am.
Yes, 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 sir.
Sir, good morning, sir. So these drugs are applicable for all type of variant coronavirus, like Delta, Delta plus virus.
Sadi, can you please repeat the questions? I could not hear very clearly.
Sir, these drugs are applicable for all type of variant coronavirus, like Delta plus Delta virus, Delta coronavirus.
No, when you thank you for the questions. When you talk about the vaccines, then I can answer, because the vaccines are targeting the spike protein where you have the mutations. Okay, the mutations, the vaccines, the vaccines have the different effectiveness against the Delta variants. When you think about the Covishield, it was reported like it shows only 60 to 68% effective against the Delta coronavirus. It was said to be, yeah, but I don't know whether we have the real results. But when you think about the small molecules, they are targeting the viral enzymes where you have a minimal chance of the mutations. Okay, that is the advantage, that we are targeting the viral enzymes. When you're targeting the viral protein, viral spike protein, then the vaccines has always the resistance problem, that because the virus keeps on developing the mutations. But in order to avoid this problem, then you really need to think about developing the antiviral drugs which are targeting the viral replications. So you take the very specific enzymes that are key in the viral replications. So when you target this viral proteases, then you will have a minimal chance to develop mutations. In this case, it will be effective for a long time.
Okay, sir. Thank you, sir.
Thank you very much. Is there something more?
Good morning, sir.
Hello.
Sir, I am Karthik Raja from Licensed Bharadas University, sir.
Yeah.
I am currently at final year, sir. Is it possible to do my final year master thesis at Germany in this situation, sir?
Yeah, you can try. Of course, there is nothing impossible. You can try. You have to approach the people. I don't know how to approach. You have to approach the people where you want to do the masters, yeah. And what is your team? Yeah, this I think it's very simple way to approach because you get the information every information in the internet. You just get the email and just ask the professor, you are interested that I want to do the master thesis. How can I continue? And you can propose your interest, and if they impressed with your, then probably they will take you. But it depends on your interest and their interest. But it is possible. Thank you.
Thank you so much, sir.
Yeah. Yes, I hope this is it.
Yeah. Thank you, sir. Thank you very much for your nice presentation.
Yeah, thank you very much. Yeah, okay. I wish you all the good. Very good day and successful webinar event. Yeah. Thank you so much.
Thank you, sir. Thank you very much.
Yeah. Bye.
Now I request Dr. Ramya ma'am to introduce Dr. Yamuna ma'am.
Okay, ma'am. Very good morning to one and all present here. It's my immense pleasure to introduce our chief guest, Dr. E. Yamuna, Assistant Professor, Department of Chemistry, Karpagam Academy of Higher Education, Coimbatore. She completed a PhD in Synthetic Organic Chemistry from Bardiyar University, Coimbatore in 2011 and postdoc studies at National Central University, Taiwan from 2012 to 15 and 2017 to 18. The current research focus is on synthesized dyes for dye-sensitized solar system. He has published 33 more papers in reputed national and international journals, and also more number of papers are presented in national and international conferences. She was a rank holder in UG degree and also a notable alumni of our college. Thank you so much for joining with us, ma'am. May I now welcome Dr. E. Yamuna to take over the session. Ma'am, please.
Thank you, ma'am. Thank you, ma'am. Good morning, all of you. First of all, I thank you, Dr. Puspaveni, for inviting me to deliver a lecture. And I would like to thank Mr. Dr. M. Lakshman Swamy, sir, for his kind words. And the topic which I have chosen is dye-sensitized solar cell, the fundamentals of dye-sensitized solar cell. Is it visible, ma'am?
Ma'am is visible. Kindly please show the slideshow, ma'am.
Okay. Is it moving, ma'am?
Yeah.
Okay. Okay. Before I start my topic, I want to tell one Tirukural. அழுக்காறு அவாவெகிலி இன்னாச்சொல் நான்கும் இழுக்கா என்றது அறம். The meaning of the குறள் is பொறாமை, ஆசை, சினம், கடுஞ்சொல் ஆகிய இந்த 4 குற்றங்களுக்கும் இடம் கொடுக்காமல் அவற்றை கடிந்து ஒழுகுவதே அறமாகும். So I am going to talk about dye-sensitized solar cell and the components of dye-sensitized solar cell and the principle of dye-sensitized solar cell and various dyes used in DSSC and their application advantages and disadvantages. And first, what is solar energy? Solar energy originates with the thermonuclear fusion reactions occurring in the sun. It represents the entire electromagnetic radiation, that is visible light, IR, and ultraviolet light, X-ray, and radio waves. This energy consists of radiant light and heat energy from the sun. Out of all energy emitted by the sun, only a small fraction of energy is observed by Earth. Just this tiny fraction of sun energy is enough to meet all our power needs. Photovoltaic cell. A photovoltaic cell, also known as a solar cell, and is an electronic component that generates electricity when exposed to photons or particle of lights. That conversion is called photovoltaic effect. The dye-sensitized solar cell. The dye-sensitized solar cell have arisen as a technically and economically credible alternative to the PN junction photovoltaic devices. It is the latest technology in solar cell, which is found in 1991 by Grazel and Oregon. This is a clean and cheap and versatile technology for large-scale production of energy. And it is based on photoelectrochemical cell, which is directly absorb light from the sun with dyes or organic layers and converted into electrical energy. This is photosynthesis. First chlorophyll sensitized zinc oxide electrode, first synthesized in 1972. This chlorophyll being the most abundant pigment that commonly found in plants, bacteria, and algae, and it plays a vital role in photosynthesis. And this chlorophyll has been experimented to function as a photosensitizer in dye-sensitized solar cell. And the study shows that this chlorophyll has the potential to be used as a new alternative energy source in the future. And we can see about the components of DSSC. So there are 4 components. The first one is a transparent conducting and counter conducting electrode. And the second one is semiconducting layer, that is TiO2, the nanostructure wide band gap semiconducting layer, and the dye molecule or the sensitizer. Example is N719, N3 black dye, Z907, or the examples for the dye, and the redox mediator or electrolyte, I3, iodine. The transparent and conducting substrate, one of the components involved in dye-sensitized solar cell. DSSC are typically constructed with 2 sheets of conducting transparent material, which help a substrate for the deposition of the semiconductor and the catalyst. And TCF for photovoltaic applications have been fabricated from both inorganic and organic material. Inorganic film typically are made up of a layer of conducting oxide, that is TCO, generally in the form of indium tin oxide, that is ITO, fluorine doped tin oxide, FTO, and doped zinc oxide. Mostly they are using fluorine doped tin oxide. The fluorine doped tin oxide FTO and indium doped tin oxide are usually applied as a conducting substrate in DSSC. These substrate consist of a soda lime glass coated with the layers of indium doped tin oxide and fluorine doped tin oxide. And this ITO films have a transmittance is greater than 80% of sheet resistance, while FTO film shows a lower transmittance, approximately 75% in the visible region. The nanostructure photoelectrode. The use of the sensitized wide band gap semiconductor TiO2 zinc oxide resulted in high chemical stability of the cell due to their resistance to photocorrection TiO2 is the most commonly used nanocrystalline semiconductor oxide electrode in the DSSC as an electron acceptor to support a molecular or quantum dot sensitizer is TiO2. Other band gap semiconductor oxide is becoming common is the zinc oxide. It possesses a band gap of 3.37 electron volt and a large excitation binding energy, that is 60 milli electron volt. Photosensitizer dye. This dye molecule is more important. The dye is the component of DSSC responsible for the maximum absorption of incident light. Any material being dyed should have the following photochemical and electrochemical property. First, the dye should be luminescent and the absorption spectrum of the dye should cover UV visible and near IR region. Next, the HOMO should lie lower than that of the redox electrolyte. And the periphery of the dye should be hydrophobic and to enhance the long-term stability of the cell, as it results in minimized direct contact between electrolyte and anode. And to avoid the aggregation of the dye over the TiO2 surface, the co-adsorbent like CDCA or chenodeoxycholic acid and the anchoring group like alkoxysilyl and the phosphoric acid group and the carboxylic acid group were inserted between the dye and the TiO2 surface. This result the prevention of dye aggregation. So, and this limits the recombination reactions between the redox electrolyte and the electrons in the TiO2 layer. This is the example for dye sensitizer. Mostly people using N719 dye and black dye. Then electrolyte. Electrolyte containing I minus I3 redox ion is used in DSSC to regenerate the oxidized dye molecule. This will complete the electric circuit by mediating electrons between the electrode and the counter electrode, the nanostructure electrode and the counter electrode. The cell performance is greatly affected by ions, conductivity in the electrolyte, which is directly affected by the viscosity of the solvent. The redox electrolyte need to be chosen such that the reduction of I3 ions by injection of electron is fast and efficient. And the working principle of DSSC. The working principle of DSSC involves 4 basic steps. First, the dye molecule becomes excited by light. Next, the dye injects an electron very rapidly to the TiO2 photoanode, this TiO2 photoanode, and the dye is oxidized in this process. So the dye is oxidized. In the first part, the dye is oxidized. Then the electrons are transported through the TiO2, semiconducting TiO2 layer, move through the load and eventually reach the counter electrode. At counter electrode, normally platinum, they will use platinum. The electrons reduce the redox mediator located in the electrolyte of the DSSC. Redox mediator diffuses to meet and regenerate the oxidized dye molecule. The work of the electrolyte is to regenerate the oxidized dye molecule. the electrons in the counter electrode in the electrolyte, the electrons in the electrolyte. Thus, the dye regeneration of the ground state of the dye molecule takes place due to the acceptance of the electrons from I minus ion and gets oxidized to I3 minus ion. And then DSSC preparation, how to prepare a DSSC plate. First, a piece of a transparent glass that conducts electricity is used as an electrode in the solar cell. It is taped down with the conducting side facing upwards using two pieces of tape. The gap between the tape is covered with a paste containing small particles of TiO2 using a process called Dr. blading. A bob of paste is put on the glass plate and the surface using a glass rod. The tape is removed and the electrode is heated at very high temperature. Then the electrode is put into the dye bath containing the dye molecules and left overnight. So the dye molecule attached to the surface of the titanium dioxide particle with a chemical bond. So this is dye molecule. It will attach the surface of the titanium dioxide particle with a chemical bond. And this process is called sensitization, which is why these solar cells are called dye-sensitized solar cell. So this is the dye we are using and this is the surface of TiO2. Next, when the electrode is removed, the TiO2 is now colored, which allows it to absorb sunlight better. So another piece of conducting glass is used to make other electrode of the cell. Two holes are drilled and then platinum metal is sprayed on one side to help the cell conduct electricity. The two electrodes are now put together with a square of easy melt plastic. After that, when heated, the plastic melts and glues the two electrodes together. Then the cell is filled with electrolyte liquid by injecting it through the drilled holes. A piece of strictly tape then seals the holes and stop the electrolyte leaking out. Then the solar cell is now complete and then can be tested in the sun. So inside the cell, this is a dye molecule and this TiO2. So a particle of light called photon hits the dye molecule. This gives an electron enough energy to escape the molecule and move to the titanium dioxide nanoparticle. When this happens, a hole is left behind. The mediator, for example, iodide, in the liquid electrolyte filled the hole. The electrons travel in a big circle. And create an electric circuit which powers a device. In comparison with the silicon photovoltaic cells, dye-sensitized solar cell exhibit some specific advantages, including easy fabrication procedure and low manufacturing cost and compatibility with the flexible. In spite of the advantages from the industrial point of view, the efficiency and the long-term stability of DSSC system have been subject of concern during the past years of development of this technology. In order to solve these problems, numerous research efforts have been devoted to the engineering and the manufacturing of devices that could meet the standard of the photovoltaic cell. But the low efficiency and the stability have remained as the major challenge in the development of dye-sensitized solar cell over the past 25 years. So these focuses on the obstacles and restrictions in the fabrication and the commercialization of DSSC. This is dye sensitized solar cell model, various colors of series. And this is also leaf shaped transparent DSSC with the 4 colors. These are all the dye. So different dye have a different color. And applications of DSSC. Because of the physical nature of the dye sensitized solar cell, inexpensive, environment friendly materials, processing and realization of various colors, power window and singles or prospective applications in building integrated photovoltaics. And the flexibility of the dye sensitized solar cell models open opportunities for integrating them with many portable devices, baggages, gears or outfits. And the lightweight of the flexible dye sensitized solar cells are attractive for applications in room, outdoor light, powered calculators, gadgets and mobiles. In power generation, dye sensitized models with efficiency of 10% are attractive choice to replace the common crystalline silicon based models. And this is a DSSC panel installed in Newcastle, Australia. And this is a flexible DSSC based solar cell model developed by Dysol. And this is a jacket commercialized by G24i. And this is self energy converting sunglass, quite simple. The lenses of the glass have dye solar cell and collecting energy and making it able to power your small devices through the power jack at the back of the frame. So in short, compared to silicon based solar cell, the dye sensitized solar cell are low cost, ease of production, and their performance increases with temperature and possessing bifacial configuration. Advantage of our diffuse light, have transparency for power windows. Color can be varied by selection of the dye. And DSSC shows a higher power conversion efficiency than silicon based solar cell. It is believed that nanocrystalline photovoltaic devices are becoming viable contender for a large scale future solar energy converter. So the search for green sources or generator of energy is considered one of the priority in today's society and occupies many policy makers. And thank you for listening. And this is just for fundamentals of dye sensitized solar cell. And in my next topic, I will tell about the metal-free organic dye sensitized solar cell and the efficiency of the dye sensitized solar cell. Once again, I thank you, the organizer, convener, and the institution management for providing me the wonderful opportunity to join with you. And thank you all.
Thank you, ma'am. Thank you for your giving nice presentation and to giving for the that is the introduction of these solar cells. Audience, if you have any queries, kindly ask your doubts. Thank you, ma'am.
Okay, ma'am. Thank you. Thank you so much.
Thank you, ma'am, for your section. Gratitude is an exquisite form of courtesy. I request Dr. A. Pushpavani, ma'am, to deliver the vote of thanks. Ma'am, please.
So, good morning to all. Respected dignitaries, faculty members, and students, I express my sincere gratitude to our beloved President, Dr. M. Arachami sir, for his encouragement and support to this academic program. I express my hearty thanks to our Director and Secretary, Dr. C.A. Vasiki ma'am, for the constant support and guidance which she has extended to all of us for this program. I thank our Principal, Dr. M. Lakshmanaswamy sir, for the constant motivation and guidance that he extended to this international webinar and for delivering the presidential address. I am grateful to our Dean Academics, Dr. S.R. Madan Shankar sir, for his motivation during this international webinar. I express my sincere gratitude to the guest speakers present here who accepted the invitations immediately besides their work. I would like to thank Dr. Tanihamale Pillaya from Germany who gave his vital time from his busy schedule to grace this meeting. And I extend my heartfelt thanks to Dr. E. Amuna from India for investing her valuable time from her busy schedule and sharing her knowledge with us in the webinar. Once again, I thank you, ma'am. I take this to extend my heartfelt thanks to the participants from the various institutions for their support and cooperation. I express my gratitude to our beloved HOD Dr. K. Kalpana Devi ma'am for supporting me to organize this international webinar. I record my sincere thanks to my colleagues, Dr. A. Amudavalli ma'am, Dr. V. Ramya ma'am, and Vadivelan sir. I record my sincere thanks to my dear students for their time and support. Thank you for joining with us. Once again, I thank for all us. Meet you all in the next academic meet. Thank you, one and all present here. Just I posted the feedback in the chat box. Kindly fill the feedback and receive your certificates. Thank you.
Thank you, ma'am.
முடிஞ்சிருச்சு வர